cardiovascular. I realize I talk loud, maybe that's why Professor Carletto closed his door. So we've talked about blood flowing through the cardiovascular system, but what we need to talk about now, instead of just moving blood, we need to hone in our focus specifically on not only the lungs, which are going to provide oxygen and a means to remove CO2 from the body, but we also need to focus, which is going to be the latter half of our of this lecture, is how we're going to have gas exchange taking place with red blood cells and within the capillaries. So that's where we're going to divide that up. We're going to talk first about the anatomy, of which I will kind of just speed through, just give you the gist of it, which you guys can read on your own. And then we're going to talk about the volume of air moving in and out of the lungs. And then we'll look at gas exchange taking place in the tissues, as well as that of the lung. So we're going to go into the mechanisms behind that, everything from the tissue layer, down to the cell layer, down to the molecular layer of how hemoglobin is going to work. So with that, looking at gas exchange, we're going to take that cellular respiration mechanism or equation back into place again, looking at glucose, oxygen, and converting that into CO2 and H2O with the process of producing 36 ATP per glucose molecule. So with that, looking at our input, our reactants, as well as our products, we know that O2 is used at that last step of the electron transport chain. We're going to reduce oxygen reducing oxygen in order to create our ATP. And then in that byproduct that we saw, all the different areas where during glycolysis, during the electron transport chain, as well, during the Krebs cycle, we saw the process of CO2 production. So we're going to need to be able to supply O2 to make sure that we can drive the equation forward and remove our byproducts of CO2. Where do we talk about the removal of water? Well, most of that we will be able to visit in our urinary lecture, but the biggest contributor of what we need to get out is CO2. So all this relates to gas exchange. How do we get O2 from our environment and then eliminate CO2 as that metabolic byproduct? So with that, we'll look at the structural division. Again, just briefly going over this, we can look at the structural division of looking at the everything from our external environment, from the nose all the way down to our functional units, which are, the functional units will be the alveoli in the lung. So we can divide that up between the upper and respiratory tract, and that's all divided by everything below the larynx down to the trachea will be the lower respiratory tract. This you can think about in terms of upper respiratory diseases versus lower respiratory diseases. And then lastly, we can look at it from a functional standpoint where conducting zone versus the respiratory zone. Conducting zone will be the mechanism in place, or the structural pieces that we have to conduct air to the respiratory areas where we're going to have gas exchange occur. So how do we get air to move through the areas into our functional units? So we'll be able to look at those units later on. And then lastly, we'll talk about the function of that conducting zone. So looking at that entire pathway, we'll move from the nasal cavity all the way out to the alveoli, of which the alveoli, again, the functional unit. Looking at the nose, you know what the nose is. There's a nose. All right. The purpose of the nose, when we see the nasal conchae, or the terminates, that allows air to flow over. And here we have tons. Not only do we see the epithelial cells for our sensory epithelial cells, but we also have tons of cilia and mucus in that area to help warm and moisten and filter the incoming air. And that's important because the air that we want to be able to inhale needs to be able to make its way down to body temperature, down to the alveoli. We also need to add in a good amount of water vapor to it. And then lastly, we need to also make sure that we filter it to remove any particulates that don't belong. The other thing is to look at, and I don't mention this, actually no, don't worry about it, it's not on the test, but sinuses are basically the air-filled chambers. There's theories as to being able to make the head lighter by keeping air-filled sinuses. It's also the reason why certain animals have bigger sinuses to cause resonation of the voice, but not that important in terms of respiratory function. 3 anatomical divisions: nasopharynx, oropharynx, laryngopharynx. This is all basically the pharynx and the divisions between these before we move on to the next structure. Next slide. The larynx. Looking at the larynx, there it is. We've moved down a little bit lower. We have cartilaginous areas. The one that's of utmost importance is the epiglottis. The epiglottis is this fold that flows over in order to block, when we're swallowing, it helps block off the trachea in order to prevent fluid or food to move into the windpipe or our trachea. The next slide is just going to show us our epiglottis, and I don't think this video is going to really work. But nonetheless, here you can see when what's supposed to happen is when we swallow food, you'll see the epiglottis. As food moves over, this is a reflex where the epiglottis comes in and closes over the trachea to prevent food from flowing through. This is what it looks like right there at the back of the tongue. And this helps, as food flows over it, pushes it right back down. This video talks about the vocal cords, not all entirely important other than the true vocal cords, which are these structures right here. True vocal cords are what give the phonation, or the sound of our voice. They're all constructed mainly of skeletal muscle, as well as some ligaments that attach. You'll see that here is our trachea. We have the true vocal folds, which allow the vibrations to take place to be able to hear what you hear. And then the false vocal cords are the ones on the sides. And those false vocal cords contribute to what's called a guttural sound. It's more of a very, what's the term I'm looking for? It's a very discrete sound, but it's a sound that, let's see if I can find it. Oh no, I do have it. Mongolian chants. Of course, I can't play it if I don't have audio. But the false vocal cords create the sort of vibratory sounds. You can hear the sort of vibrato that's happening in the back. That's a sound. So those sounds, English language doesn't really use these sounds all that much, but there's other languages that utilize the guttural sounds that we're not so accustomed to. Filtration that takes place. Here you can see a image, our scanning electron microscope. We've got a histological slide with the highlighted cilia. And they've highlighted the cilia that are on the apical side of the epithelial cells. Here you see the columnar epithelial cells, of which some of the basal cells produce mucus that gets secreted out. And that mucus lays right on top of the cilia, but the cilia helps sweep to capture any particulates that get embedded by the mucus, and that cilia helps sweep out back to get rid of that excess stuff that we don't need. And so the cilia in the upper respiratory tract help trap the particles into the pharynx so that we can, and then on the lower respiratory tract, move it upwards towards the larynx so that we can essentially cough and swallow it down to make sure that it doesn't make its way down into, down to the trachea, down into the bronchioles, into the lungs itself. So this is our way of doing it. The other thing that I can mention of note that is noteworthy is that cilia is what we have that increases the surface area. So not only does it capture particulates, but it is meant to basically help protect. And cigarette smoking actually comes in and stops the cilia from moving. So that is something that is a concern for those that are regular smokers because cilia, it takes about a couple hours for them to restore their functions after they've been stopped by the effect of smoking. And that pretty much mutes the whole effect of the filtration that does take place. Lower respiratory anatomy. Lower respiratory anatomy at some point. There we go. PowerPoint is not responding. No. So let's work on our slides while this is happening. So lower respiratory anatomy. So lower respiratory anatomy. Here you can see everything below the larynx. The thing is about the trachea is it's semi-rigid in which the anterior portion is cartilaginous and the posterior is going to be made of smooth muscle. So this is going to be fairly important because right behind is the esophagus. So the esophagus is right behind it and this allows, that smooth muscle allows for the adrenergic receptors as well as the cholinergic receptors on there will respond to allow the trachea to open up a little bit more as well as to dilate or to constrict. The other thing to look at besides the structure of it is to see the different layers. You'll see once again the same layers that we saw earlier are mucosal layers which are going to produce mucus to line up the inside. You'll see the connective tissue on the outside. Submucosa, the hyaline cartilage, and adventitia. Again, not as important, but really just to contribute to the overall structure of this transverse cut. The other thing to look at the lower respiratory anatomy is that it allows for conduction. Again, part of the conducting zone that will split between the left and the right primary bronchus, or bronchi. Again, the purpose of the conducting region is to filter and moisten the air and warm the air back up to body temperature. The other area that is of importance is the carina, because here we do have sensory receptors that detect any particulates in that area. They're very sensitive to anything. So that promotes what's called the cough reflex. So there are drugs that work against the cough reflex, like Delsym, anything with dextromorphan comes in and actually will desensitize the carina to prevent coughing from taking place. Again, the bronchial tree basically breaks down left and right, and then secondary bronchi is the next split from, so if this is our left and right primary bronchus, you'll see our first split will be our secondary bronchi. And then our tertiary bronchi is again another split. We move from the terminal bronchioles to the respiratory bronchioles to the alveolar sacs. These right here comprise the functional unit, or rather, our alveolar sacs are the functional unit. But everything from here is going to be our conducting zone, so our respiratory zone. Well, everything from that up to the nose represents our conducting zone. Again, the significance of the conducting zone is to filter and moisten and warm up the air, while the respiratory zone is going to participate in gas exchange. Bronchial tree shown on the next slide over, with the lungs superimposed as well as the diaphragm. Looking at the primary to tertiary, the makeup, the tissue makeup of it, there's less cartilage and more smooth muscle as we make our way down to the tertiary. Why the smooth muscle and less of the cartilage? Less structural, but more so functional by the time we make it to the tertiary bronchial. And the smooth muscle there, again, have the adrenergic and as well as cholinergic neurons that will respond to autonomic pathways. So the autonomic regulation, the sympathetic will cause dilation. We want to increase the size of our bronchioles to allow more airflow into the lungs. The parasympathetic is going to cause constriction to reduce the amount of airflow into that area. So this, again, when looking at the process of resistance, or is all tied in, again, to the radius. We can adjust the radius of the bronchioles in order to allow more air in or allow less air in. And that takes place within certain bronchioles in order to allow for air to move in as well as gas exchange to occur in those areas. The alveoli, again, moves us directly into the site of gas exchange. And from here, these are going to see the respiratory bronchioles, and then you have these clusters of grapes, basically, like to call them. They're the pouches that form, that fill up, that allow air to participate in gas exchange. If you look at the structure of the alveoli, the cellular structure, simple squamous epithelial tissue. So flat, single cell epithelial tissue, one layer thick, and that is going to be where our gas exchange takes place. The respiratory bronchioles, for the most part, aren't really functioning other than to bring air into that area, but is still considered part of the functional unit. So while I mentioned the alveoli are the functional areas, the entire functional unit is what is called the pulmonary lobule. And that consists of the alveoli, the respiratory bronchioles, the the vessels that lead to that area that will provide the capillary bed to allow for gas exchange to occur in that area. What separates the functional units are the elastic connective tissue that wraps around. So this would be the bag that wraps around the alveoli, and that's going to play an important role in the way we can move air in and out of the lungs. So keep that in mind when we move on to the next section. Alveoli. Looking at the cellular makeup of the alveoli, you'll see we have type 1 alveolar cells. The type 1 alveolar cells are the actual cells itself. This is the simple squamous epithelial cells that you see that allow for gas exchange to occur. This is the most prominent type of cell because this is where we, due to the simple squamous cells, we have the most surface area. So how much surface area does it come out to? Again, depending on somebody's height, age, weight, gender, and height, age, weight, gender, and as well as race also plays a factor, gives us the size of the lung. And with that, we have about 750 cubic feet. So if I were to assume that this room is 20 feet by about 35 feet, something like that, which kind of makes sense. It's about the size of this room. That is the amount of surface area that we have for gas exchange to occur. That includes the capillaries that provide the area where red blood cells pass through, and an interface on the alveoli that allows a surface for gas exchange to take place. So here we're going to see fresh O2 in this area that will move its way into the red blood cell, and then we have CO2 that's being carried by the same red blood cells moving out into the alveoli. The next thing is to take a look at the type 2 cells, which are simple squamous cells again, but they are specific excretory function. Why? Because they secrete surfactant. Surfactant is something that's important to prevent the stickiness of the alveoli. So these tissues, since they're so close together, would normally want to stick together. If you press the alveoli together without surfactant, you'd have a difficult time trying to separate those back up to fill back with air. A good example would be to take a wet Ziploc bag. Take a Ziploc bag and fill it up with a little bit of water, take out all the air, and look how they stick together. Water, which surrounds, again, these cells again, have a tendency to have very high surface tension and strong cohesiveness. So they want to stick together. Surfactant does the exact opposite of that, and it makes it a little bit looser, and it breaks apart those water molecules to reduce the tension that holds them together. So this is something necessary to allow that the alveoli to be able to properly inflate. And something that they also do with babies as well. If premature babies, sometimes, actually premature babies typically will not produce enough surfactant, so they do, I believe, have a spray that they can use in order to get it back into their lung. It's an artificial surfactant that they can initially use to open up the alveoli to allow for their own natural production of surfactant by the type 2 alveolar cells. Macrophages are the, again, part of the immune system. The alveolar macrophages are all part of just taking away things that don't belong. Any debris, any diseases of any kind, they come in and work to take all that away. Blood supply to the lungs. Pulmonary circuit. So there's two parts of it, where we're going to see the pulmonary circuit has the delivery of blood flow into the capillary beds that supply the alveoli. So the pulmonary arteries are going to deliver deoxygenated blood. So that's moving again from the right ventricle down the pulmonary trunk. Pulmonary trunk will split into the pulmonary arteries that will deliver blood to the lungs. The capillary bed where the gas exchange occurs, and then the pulmonary veins, which return back to the left atrium, carrying oxygenated blood. So again, arteries carrying deoxygenated blood, veins carrying oxygenated blood in the pulmonary circuit. Bronchial circulation is just something very similar to what we saw with our coronary circulation. This is our way of delivering oxygenated blood to the bronchi and the bronchioles. Lung divisions. Three different divisions of the right lung, 2 divisions of the left lung. Apex of the lung, and then the bottom, the base of the lung. The base, you'll see, is kind of curved due to the diaphragm. Great. Next slide. Picture of the lungs. Next slide. Pleural cavity. Pleural cavity is basically the cavity that surrounds the lungs. So the lungs aren't just sitting amongst the ribs and the intercostal muscles or anything like that. They're actually covered by pleura. So the pleura is actually double layered. It's got one layer on the inside, one layer on the outside, of which the visceral layer is the one that actually covers the lungs themselves. The other layer, which is the parietal layer, is the one that actually lines against the thoracic cavity, right up against the ribs, and then the diaphragm itself. So the purpose of that pleura we'll be able to see on the next slide over. So in between, you're going to see the space in between the ribs and the lungs. This is our pleural cavity, and it's important in ventilation because it serves to, it's filled with a very small amount of fluid to allow friction, or to rather reduce friction, and allow the lungs to slide open and closed. So as the lungs inflate and deflate, this helps provide a slippery surface for that to take place. Again, the lung tissue is very delicate, and this is going to help protect. And we'll be able to see the mechanism in terms of gas pressure, or the air pressures that take place. The next thing to take a look at is the, give me a second, looking at atmospheric pressures, we're going to look at the composition of air. So we're going to take a step back from the anatomy and now move towards some of the laws that we need to look at. Give me a second and see how far I can move forward. We have three lectures, right? This is the first. We have next Tuesday, and we have next Thursday, and then you have
next week, whole week.
Which one? Oh, this is the entire respiratory lecture.
Oh, okay.
Yeah. 2 more days.
Give me a second to kind of plan this out in my head. Next week. Next week. Ventilation. Once I get to this.
That's it.
We have a quiz next week. Do we have any labs next week?
No, we don't have quizzes.
One
No,
more.
no, no.
No more
After- Long volume, yeah, long volume, right?
Yeah.
Oh, we're doing good on time. All right. Okay, so pleural cavity, as I mentioned, in between, great. Atmospheric characteristics. Let me cover a couple more slides. I'd like to at least get to ventilation and the mechanisms behind inhalation, exhalation, and we'll be good from there. All right, so the atmospheric characteristics. Now that we look at the volume of air, we were surrounded by air, obviously. We want to be able to not only look at the pressure of air, but we also want to look at the composition of air as well. So in the composition of air, we're basically taking a sample of air and finding out what percentage of that is going to represent the gases that are in air. And so looking at what exactly is air, air is basically all the different molecules. When we're looking at nitrogen, oxygen, carbon dioxide, water vapor, carbon monoxide, a bunch of other stuff that's actually dissolved in air. The biggest bulk of what air is are just nitrogen molecules, 78%. Oxygen is about 21%, carbon dioxide about 0.04%. And that almost adds up to a little bit over 99% of what air is comprised of. So if you think about, if we were to sample air and we think that our air is rich in oxygen, yeah, it's rich in oxygen compared to other planets, but we still have nitrogen, quite a bit of nitrogen in there, which is an inert gas that we don't really use for the purposes of gas exchange. But oxygen is 21%, carbon dioxide 0.04%. That at least gives us an idea of what the percentage of gases are, no matter what pressure we're looking at. So if I were to go back up and I were to take open up an airplane and take a sample of the air there and measure the percentage of O2, it'd still be 21%. I could go up to the very top, the stratosphere, and measure the air and it would be 21%. I can go down to sea level and measure it and it's 21%. The composition of our gases anywhere on this earth is always going to be about 21%, well, assuming up in the air somewhere. Now pressures are going to be different. And again, going back to what I was describing earlier, we have nitrogen, we have oxygen, we have carbon dioxide, most of it being nitrogen and oxygen. And those molecules are bouncing around. They've got a bunch of kinetic energy. And if I were to measure a container that they were in, the rate of them bouncing around this entire container is going to exert a net force on that container. And that net force is basically pressure. Pressure is a force being applied over that general area. And so if we were to take a look at sea level, which we call 1 atmosphere, at that pressure we have all those molecules bouncing around. And as we move further and further away, we'll see that pressure start to drop. So altitude is going to be a factor. The higher up we go, the less total number of molecules that are up there bouncing around, therefore less pressure, but the composition is still the same. We still have the same percentage of oxygen and nitrogen molecules up there. So this is why it's important to measure pressure, why we have such a concern about the atmospheric pressure and the day-to-day variations that take place. From a side note, that's going to influence the movement of our air patterns and weather patterns and so forth. All of our pressures are measured by a barometer. Just like you read about our baroreceptors, barometer is related to pressure. And the idea behind pressure, just the same way with blood, whether you're talking about air, you're talking about fluid, they're all mediums that almost exhibit the same type of movement ideology. Air moves from high pressure to low pressure. If I were to have something with really high pressure in here, and I were to open it up, the second I opened it up, you would hear that hissing of gas moving from inside to outside. Or if the other way around, if this were a vacuum and I'd open it up, again, you hear the hissing because we get atmospheric pressure trying to make its way into the can. So this starts to lend itself into the whole idea of how ventilation, which is how our lungs move in and out, which we'll talk later on. I've got several slides that talk about that. How do we get air in and out of the lungs is through pressure that we can generate. And what's going to be generating that pressure? Well, those are the muscles and that are going to be involved in expanding the lung and contracting the lung to get air in and out. So that is pretty much a quick summary of how those work. The other thing too that I didn't quite mention about other than altitude is temperature. Temperature is going to raise the kinetic energy of these molecules. And as they move faster and faster, that's more pressure on the container they're on. So if I were to, for instance, take a pot, like a cooking pot, and I leave it there and I close that pot, and I were to raise up the temperature, I notice that the pressure will start to increase inside, because again, the kinetic energy would lend itself to bouncing around in there and increasing the amount of pressure from there. So again, all that ties into our air mechanics that we'll talk about later. So the origin of the barometer. We had ideas. Basically, what this comes down to is how do we get air to move due to the pressures that are exerted by air pressure versus pressures that we exert. Let me give you a good example. How do you get straws to work? You ever think about that? You take a straw, and what are you doing? Do you think you're sucking up fluid through the straw? No, technically you're not. Actually, what you're doing is you're creating a negative pressure in respect to that of air pressure to cause fluid to move from a high pressure to a low pressure. So if I'm creating suction with my mouth, that's causing fluid to move from air pressure out, moving it up the straw into from an area of high pressure to low pressure. Now, the concept of the barometer came about when back in the 1600s, Galileo thought about the whole thing of being able to draw water from a well. So if you imagine way back when they had a well and they had a pump, and they were wondering, how do you get water to move against gravity? And he postulated that it was this whole concept of the force of a vacuum that he's creating in order to do so. But Torricelli was a mathematician and a physicist from the 1600s, came up with a different explanation. And he thought that it wasn't necessarily the vacuum that was exerted, but it was the pressure of air. And so he took this example where he took this really long glass tube. And back then, it was really easy to make these glass tubes. We've been making glass for a very long time, and very, very cheaply, too. He took this tube, and it was, I believe, open-ended on one end, but that's not really that important. He took this glass tube and stuck it in a vat of water. And he wanted to be able to see what would happen with this as he filled it up with water. This was a very, very, very long tube that he used. And what he noticed is that the water would move its way up the tube. And so you saw the effects of the cohesiveness of water, but also the adhesion along the tube. And it makes its way up. And as it makes its way up, he again theorized that it had something to do with the pressure being exerted on the surface of the water that's exposed to the air, pushing it through and forcing the fluid back up. And he noticed it fluctuated quite a bit. The fluctuations on there varied from time to time, and he couldn't really figure out why that was taking place, but it had something to do with the atmospheric pressures. He noticed on hotter days, we noticed that pressure going up a little bit more. On colder days, the pressure going down a little bit more. But more so, it still hovered around a certain number. And with water worked, but the amount of glass tubing he needed to use was somewhere on the magnitude of 15 to 20 meters long. So he thought, well, let me use a different fluid. Let me use something a little bit thicker, obviously a lot more dense. So he chose mercury. He did the same experiment, used a smaller tube, and he filled this with mercury. And he filled it up, and once again, watched the same fluctuations that took place. And how high did it go up? On average, he noticed at sea level, it went up about 760 millimeters up from the very base all the way up to the very top. And again, fluctuations that took place between 770 to about 750 millimeters of mercury. And so what he was able to prove at that point was over and over, these repeated experiments showed that the atmospheric pressure, the average pressure that caused that mercury to move up was 760 millimeters. Now he repeated the same experiments by going up in altitude and doing the same thing, and he noticed, well, the pressures are actually lower. So therefore, with that, he was able to use this measurement, 760 millimeters of mercury. So if you saw me doing that with our pressures, the same pressures, millimeters of mercury. It's a standard that he was able to establish. Again, his name was Torricelli. And so we named it, in response to him, we called it 760 torr. And 760 millimeters of mercury comes out to 1 atmospheric pressure, which is what the unit that's also used as well. So these are used interchangeably, whether you're saying torr, millimeters of mercury, and then 1 ATM, which is atmospheric pressure, is what it comes down to. So that gives us the idea of where that term pressure, or rather millimeters of mercury, came from. So looking at that, now that we know how the pressures are derived, let's take a look at our common gas laws. Common gas laws, and then, yeah, okay. So common gas laws, there's 3 laws that we need to account for that talk about the mechanisms of volume as well as composition of air. Boyle's law. Boyle's law is that volume and pressure are inversely related. And that comes down to taking that same container on here, taking that same container, and pushing down on that container with some form of piston. We're decreasing the volume, but then again also increasing the pressure contained within. Okay? And then doing the exact opposite, where we can increase the volume and therefore decrease the pressure contained within. And that should make sense, because here in this image you can see the different molecules that are all embedded within, and by decreasing the volume, compressing that volume, keeps the same molecules in there, and they're bouncing around exerting more force, therefore increasing the pressure. Another example is to think of if you have like a water bottle and you're squeezing it. If you're squeezing it, you're going to see the same pressure increasing because you're compressing the volume. The next thing is to look at Dalton's law. Dalton's law is basically saying if you were to take the pressure, let's say 1 atmospheric pressure, or in this case 760 millimeters of mercury, that's for the pressure of all molecules in that air. All we need to do to figure out the partial pressure of each gas, or exactly whether we're looking at O2, CO2, or looking at nitrogen, we know that of all those molecules in that air sample, if I got 100 molecules of gases in there, 21 of them are going to be O2, 78 of them are going to be nitrogen, and then 0.4 of them are going to be CO2. So all we really need to do to take partial pressure is to take the pressure of air and times that by their composition. So looking at 760 millimeters of mercury for O2, or for all the gases, I can say that 21% times 760 comes out to 159.6 millimeters of mercury. So that's O2. So about roughly 160 millimeters of mercury. And again, that's just those O2 molecules. That's the net pressure of just those O2 molecules on that surface. And when you add up each of the gases contained within, it comes out to 760 millimeters of mercury. Henry's law, on the other hand, is basically saying if you increase the partial pressure of a gas, let's say for O2 I go from 160 millimeters of mercury and I'm going to increase the partial pressure. Okay? And I give that in contact with the solution. I'm going to, that solution is going to be more, that solution is going to have more of that gas dissolved in it. Let me give you something that's applicable to help you understand. Do you ever wonder why beer or soda uses CO2? Or why craft beer places are using nitrogen? Why don't you use oxygen? Well, it's because oxygen doesn't really dissolve well in the fluid. CO2 actually does. That's why carbonated beverages, if you pop open a soda, you notice not only is the pressure high inside, it's filled with CO2, and that high pressure of CO2 inside there allows for more of that CO2 to dissolve. So try this at some point. Take a look at the glass bottled sodas, and take a look inside, and you can't really see the bubbles in the soda. But once you pop the top, you're going to release some of the pressure, the partial pressure. It's mostly CO2 in there. You're going to release pressure from that bottle, and then you'll see the bubbles start to form out of that soda. So that's all you really need to do to get more CO2 in there. You just got to put it in there, and you just got to add more CO2 and highly pressurize it. Increase the partial pressure, and you'll get more gas dissolved into that. So the question is, how does this apply to physiology? Here you're talking about soda. How does this really apply? So think about where we see CO2. We produce CO2 as a byproduct. So the mechanisms as to how we hold CO2 in the blood is going to be a lot easier because we have CO2 just dissolved into the blood. More of that dissolved than O2 will. So that will change the mechanics as to how we get O2 out and CO2 in. And with that, let's take a look at our respiratory system components. We'll go through the we do ventilation, we do gas exchange. We're doing really well. Okay, great. So I'm just going to do a couple more slides and I'll get you guys to do your labs. So with that, far back am I going? Did I lose myself? There we go. Pulmonary ventilation, the respiratory system components. We have to look at ventilation, pulmonary ventilation, with just strictly the movement of air. We're not looking at the composition of the air, we're looking at the movement of air. How do we get in and out of the lungs? Pulmonary ventilation is the movement out from our atmosphere out into the lungs. This is the mechanics of how we breathe. The second and third, pulmonary external respiration and tissue internal respiration, all tie down to gas exchange, which are going to be utilizing our red blood cells, which are going to contain hemoglobin, to allow oxygen uptake from the air and CO2 release into to our air, to allow gas exchange taking place externally, away from the tissues, and internally within the tissues. So as we exchange gas on the surface of the lung, well, specifically in our pulmonary lobules, specifically within the alveoli that are surrounded by the capillaries, here in tissue respiration, we're going to see the same capillaries that are delivering blood with the pressure, are allowing gas exchange to take place. Remember what we said about our red blood cells. Red blood cells travel single file through the capillaries, so they will allow for that interface between the tissue and the capillary to allow gas exchange to take place, and same as what happens in external respiration. Now, some terminology I'd like you guys to get familiar with is, you know what, no, that's cool. We'll end there. We'll revisit a little bit of this on, today's Thursday, right? Yeah, next Tuesday. We'll stop there. Actually, we made a good segue, and we'll get into our lab. So our lab shouldn't take too long.